All right, this is our week about DNA and how it functions. We're not going to dive into hereditary here, how genes are passed down and that kind of stuff. No Mendel, no Punnett squares. But we are going to focus on what DNA is, how it's used to make proteins. So that's our big theme for this week. How is DNA used to make proteins? Okay, ready? We're going to start first with an overview of the DNA molecule itself, a little bit more detail about how DNA is arranged in cells, and then talk about the process of gene expression, transcription, and translation. And then we will end with the process of how genes are regulated. How do we turn on and off genes and that kind of stuff. Let's dive in. All right, so the genome. You may have heard this term before, right? Genomics, genome studies. It's kind of a buzzword lately. The genome just means all of the DNA an organism has. Its entire set of DNA, that's all it means, right? But different organisms will have different amounts of DNA. They'll have that DNA maybe arranged in a circle, like in bacteria, or in us, it'll be in chromosomes that are linear pieces, right? We're going to focus on DNA in humans and what that looks like. So our genomic DNA is its entire set of DNA, all the DNA it has. And for every cell within a multicellular organism, all of our trillions of cells that are human cells in our bodies, they all have the exact same set of DNA. They all have 46 chromosomes, so 23 chromosomes in pairs of 2, okay? So every single cell, your eyeball cells, your muscle cells, your bone cells, they all have a total of 46 chromosomes. They all have the exact same type of DNA. The difference, getting ahead of myself, but the difference is different cells will turn on and off different genes, so that way they become a skin cell versus a bone cell versus an eyeball cell, right? But so we have about 3.2 billion base pairs in our across our 46 chromosomes, roughly. So a base pair is A and T bonded together, or G and C bonded together. Those two bases of DNA bonded together, that's a base pair. So about 3.2 billion of those, roughly, in all of our DNA. And in humans, we arrange, well, all eukaryotic organisms arrange our DNA into linear pieces called chromosomes. So in our nucleus of our cell, we'll have linear pieces of DNA. Each one of these is a chromosome. And again, they come in pairs. I always say homologous pairs. Okay, so we have a total of 23 pairs of chromosomes. So a total of 46 overall, coming in pairs like this. And when DNA comes in pairs like this, when it has, organism basically has 2 copies of all of its DNA, we say that organism is diploid, or it's a diploid cell. So di meaning 2, and ploid referencing the number of chromosomes. So we have 2 copies of each chromosome. So we are diploid organisms. Okay, let's dive into more what are those diploid chromosomes about? What are those homologous chromosomes? What is happening here? So during reproduction, sperm finds egg, they fuse together, right? The egg and the sperm are each carrying one version of each chromosome. They fuse together in fertilization, and that's how we get back to being a diploid organism, right? So you've inherited one chromosome from your father, one chromosome from your mother, okay? So each chromosome has the same type of information. So let's say these are both chromosome number 1. This is chromosome number 1 pair. You got this chromosome number 1 from your father, this chromosome number 1 from your mother. They have the same basic type of information. They're gonna have genes for, let's say maybe this R gene is if you can digest dairy or not, right? If you can break down lactose. But they're gonna have the same type of information at the same location on each chromosome. So each gene for a trait is gonna be found in the same place on both your mom and your dad's chromosome, and then chromosomes that you inherited from your mom and your dad. But they could have different forms of that gene on each chromosome. So like, let's say down here, let's say maybe this is the gene for curly versus straight hair. I don't know. Let's say from your mom, you got a curly hair gene, and from your dad, you got a straight hair gene, right? So it's still giving us that same type of information, the texture of your hair, let's say, but different forms of that information. Okay, That's how we kind of end up being kind of a mix between our parents, is we're going to inherit the same type of genes from our parents, but we might get different forms of that gene. Okay, so I've accidentally used some vocab here without explicitly defining it. A gene, like these that are highlighted here in these bands that are given letters, that's just a piece of DNA that codes for a specific protein. So a gene is a piece of DNA that codes for a specific protein. Each gene in our genome has a certain location where it is located. So in every single person, usually, they'll have the same genes in the exact same location within their entire genome. And we call that location where a gene lives, its loci, or locus for singular, or gene loci for talking about multiple genes. Okay, so it just means location. Okay, locus just means location, where we find that gene within the 23 chromosomes that the 23 pairs of chromosomes that humans have. So each gene has a specific location, a locus, where it lives on a chromosome, and different forms of that gene, we give it a special name. So like this one down here, we said curly and straight hair. There's still both the gene for hair texture, right? Those different forms of a gene, we call those alleles. I don't know where that one comes from, but that's the term that we use for different, like, if you have hair on your middle knuckle finger, if you don't have hair on your middle knuckle finger, those are different forms of the same gene. We call those alleles, right? Tall versus short. I'm simplifying here, but different forms of a same gene are called alleles. So this is a simplified version of a chromosome. We'll be looking at these a little bit more, and we'll be looking at them more next week when we talk about cell division. But the key here is that we have 2 copies of each chromosome, which means we have 2 forms, or 2 copies basically, of every single gene in our genome. So here's where it gets fun. The combination of genes or alleles an organism has, the types of alleles that we find in its genome, we call that its genotype. Okay, so genotype, basically gene type is what that means. So the type of DNA an organism has in its genome is its genotype. The phenotype of an organism is the physical characteristics of that organism, which are determined by the genotype. So a combination of genes determines the physical characteristic or the phenotype of an organism. And we're going to be talking about how that genotype gets turned into phenotype later in another video. But genotype determines phenotype because genes contain the recipe, the information for how to build proteins. So the physical characteristics of us, everything that makes us us, is a result of what we call gene expression, turning that information in DNA into a physical form of a protein. And that process, gene expression, that's kind of what we're going to talk about in our next video. So we can think about a genotype, DNA an organism has, the combination of alleles that they have, phenotype, physical characteristics, or traits. You'll often heard phenotype talk about traits. What traits does an organism have, right? It's like right here we have a fruit fly. It has the genotype for straight wings, so it ends up with straight wings because these genes tell the proteins in its wings to take a certain shape. This fly over here, look at its wings, it's kind of wrinkly and weird looking because it has the genes that result in its proteins taking the shape that gives it a little bit of a wrinkly shape to it. Okay, so genotype, combination of genes, combination of alleles. Phenotype is the physical characteristics or traits that are a result of that genotype. Okay, so DNA is contained in linear chromosomes in organisms, in eukaryotic organisms. We have 23 pairs in humans. We've talked about this before, but just a refresher in case it's kind of muddled back in there with all the other stuff we've been talking about. Right? Remember, DNA is made up of nucleotides. That nucleotide is just that building block of DNA. We have 3 pieces to our DNA nucleotide: phosphate group, deoxyribose sugar, and a nitrogenous base. We can swap out this base for all of these different forms, and that's what gives us the A, T, C's, and G's of DNA, the different variations of this nucleotide monomer. Do you remember what would be different if this was, so this is DNA, how could you tell if this was an RNA nucleotide? If you can't answer that right now, go back and study. But what are some things that you could look at to determine if this was an RNA nucleotide and not a DNA nucleotide? It has to do with the sugar in one of those bases. So DNA, when it's in a cell, always comes in two strands that run anti-parallel to each other and form a double helix structure. So DNA runs anti-parallel, meaning one side is basically upside down compared to the other side, based on the orientation of those nucleotides. See how this one has the phosphate groups facing up? This one has the phosphate groups facing down, okay? So they're basically flipped. They're running upside down from each other. So that's what we mean by anti. And then parallel means that they're running next to each other, but they don't ever actually touch. These two strands of DNA don't ever actually touch. So what we're looking at here, these nucleotides, if we were to zoom in on some of these base pairs, when we diagram DNA like this, it looks like they touch, but in actuality, they're running parallel to each other. Right? They're not actually touching, but they're being held together. The two sides of this DNA molecule, the two strands of DNA, are being held together by hydrogen bonds between those nitrogenous bases. So in cytosine and guanine, there's 3 regions of hydrogen bonding. Remember hydrogen bonding, this oxygen is going to have a slight negative charge. Hydrogen is going to have a slight positive charge to it, and they'll be attracted to each other. Right? This hydrogen here is going to have a slight positive. This nitrogen here is going to be slightly negative. They'll be attracted to each other. Okay, so slight charges caused from polar bonds, polar covalent bonds, is what causes hydrogen bonding. So 2 strands that are anti, upside down from each other, and parallel, not actually touching, but because these hydrogen bonds. It's not a covalent bond, they're hydrogen bonds. And then it forms this double helix shape, which just means that it kind of twists and turns as the molecule takes shape. Running along the side here, so this light blue ribbon right here, that part of the DNA molecule is a sugar phosphate bond. So we call this a phosphodiester bond. So we have a phosphate group and the deoxyribose sugar. That's the component of the nucleotide that results in what we call the backbone of the DNA molecule. So this light blue strand right here is represented by sugar phosphate. Sugar phosphate. So those are the components of the nucleotide that result in the backbone of the DNA molecule. All right, here we go. Remind yourself. Remember, how do you tell the difference between DNA and RNA? RNA, remember, we have a different sugar in RNA. DNA uses deoxyribose. RNA uses ribose. Okay? And in RNA, we'll see the base uracil replaces thymine. So if it's a sequence that has thymine in it, it's going to be a DNA sequence. If it's a sequence that has uracil in it, the letter U, it's going to be an RNA sequence. So that's how you can tell the difference between DNA and RNA. The sequence is, it has U versus T. T is DNA. And then the sugar. Ribose has an extra oxygen. Deoxyribose is lacking in oxygen. We have our antiparallel double helix. It's arranged in linear chromosomes. Humans have 46 of them, come in homologous pairs of 23 homologous pairs that they have same information but maybe variations on that information on the homologous pairs. But how do we get all that DNA in a cell? So if we were to take the DNA from just one of our cells, we were to extract the DNA from our cell, and we were to extend it out in one long strand, all 46 chromosomes in one long strand, that'd be about 2 meters. 2 meters. That's over 6 feet of DNA, one strand of DNA in every single cell. That's a ton of DNA. That's a lot of information. It's a big molecule to fit in such a tiny microscopic structure of our nucleus, right? So how do we get it all in there? How do we get all of that DNA into our cell and packed tightly into our nucleus, right? Well, how would you do it? Think about if you had 6 feet of a string, how would you wind it up to its smallest form possible? So DNA is condensed in a very specific manner, so that way it's still functional, our cells can still use it, but yet it's still able to fit inside of our nucleus. So we take our DNA double helix, right, that sugar phosphate backbone with those bases in the middle stuck together with hydrogen bonds, and then we wrap it around a protein. So you know how you can kind of take like thread and wrap it around a spindle, kind of. That's basically what's happening here. We're taking DNA, so this kind of double loop structure right here is the DNA molecule, and we're wrapping it around a protein. Okay, so we're wrapping it around a protein, and we're going to wrap it around some more proteins, and wrap it around some more proteins. These proteins that DNA is wrapped around are called histones. It's a very special type of protein that DNA wraps it around. It's called a histone protein. Then we take many pieces of DNA that are all wrapped around histones. So this is one long strand of DNA. Each piece of that strand is wrapped around a histone. We usually don't need to remember this, but if it helps, around 200 base pairs roughly of DNA get wrapped around one histone. So we wrap it around a histone, and then we coil it again. So we start to wrap that piece of DNA that's wrapped around a histone, we start to coil that again. And then we have more coiling. So you know when you take like a string or something and you start twisting it on either end and it starts to like kind of wrap up on itself in the middle? That's kind of what's happening here. We're taking DNA, we're wrapping it around a histone, we're wrapping those histones together, and then we're wrapping all those histones that were wrapped together, we're wrapping them together again. Finally, we call these condensed pieces of DNA chromatin. So this section of DNA right here that's been condensed, this is one chromosome right here. It's been replicated, but whatever. This one section of DNA right here, it has many, many pieces that are all connected together like this. And this little ring, this little loop right here is basically all of this. Okay, so kind of hard to visualize, but this would be all looped up DNA, and then each of those is wrapped up. So that's how we can take 6 plus feet of DNA and put it inside our chromosomes. Big idea here that we're wrapping it around histones, and then we're hypercoiling it, supercoiling it, so that way it's nice and compacted into a structure that we call chromatin. So when it's condensed, when DNA is condensed, we call it chromatin. That's kind of the big thing I wanted you to get out of all that.